Nice to meet you. I'm Ruthie. Today I'm going to be talking about biomaterials as UI, designing new user experiences. So for those of you in the back, this is me. I wore the same dress, so you can see me. You can recognize me later. A little bit about me. I live in New York City. That's my Twitter handle. If you're ever in town or if you live in New York, tweet at me. I'll buy you coffee and we'll hang out. I work as a software engineer at a company called Warby Parker. We're an e-commerce company. My colleague is over there. We sell eyeglasses and sunglasses, and so I'm a software engineer on the systems development team there. And so I work on things such as software for logistics, supply chain, retail operations, customer experience tools. And I was thinking about how the end result of all this software that I'm working on is actually this physical product, eyeglasses. And so I was curious in understanding how people are extending programming today to physical materials. And so I started to learn about this space, and I got to this field of biomaterials, which are materials that are either derived from nature or synthetically produced based on materials found in nature, or some kind of combination of the two. And the reason that they're an interesting type of material to program is that they already have forms of programming happening within them through their biological systems, whether that's feedback loops or repair mechanisms or different life cycle events such as reproduction and decomposition. And so people have been exploring ways to extend those and to harness different naturally occurring attributes and traits in these biological systems to program with these materials. And the reason I think that it's really important and interesting to learn about as software engineers is, first of all, most of the interfaces that our users interact with are not the ones through which they experience our software on. There's a lot of materials that people touch and use every day that we can design new experiences for. And as software developers, we design for the capabilities of the interfaces that we build for, whether that's the haptic quality or pedometer or gyroscope or camera of a smartphone or the conversational user interfaces of an Alexa or Google Home or the form factor of a VR headset. So hopefully by learning a little bit more about what's possible, you can start to imagine new types of experiences you can build and extend to different materials. So what if we could create clothing that responded to heat and humidity changes in our body by opening up and helping our body return to equilibrium and then closing the flaps when we cool down? Researchers at the MIT Media Lab's Mediated Matter group, along with colleagues at the Royal College of Art in London and Zhejiang University in China, asked themselves this question. And this would be quite hard to do with traditional sensors and mechanical and electrical engineering, making the fabric open up and close. But then they found that this actually happens a lot in different biological systems. They honed in on this soybean that has, when it's fermented, it has this bacteria called Bacillus subtilis that basically changes its form in reaction to environmental changes. And so basically nature is full of all kinds of sensors and actuators, things that trigger motion. And so they're able to utilize that and to utilize a new type of material engineering called bioprinting to create films. They then put on these fabrics and we're able to make them respond to changes in the body. So I can show you what that looks like on a closer scale here. You can kind of see the biofilms. And then this is a zoomed in picture of the bacteria that they utilized. And this is a picture of the bioprinter that they used to print these films. And this is them growing some bacteria in a lab. So some of the advantages of using a living system as a sensor and actuator are it's safe, there's no wires, it's edible, it reproduces easily, and you can use that as another design element. And also, you can use the reproduction as a design element. At the same time, there are also some challenges. For any of you who've done biology experiments, you know that sometimes you put the same inputs and you get different kinds of outputs. And so trying to make the results of their production of the bacteria in the lab get the same results each time has been challenging for them. Beyond that, they also have been trying to play around with their ability to configure exactly the reaction that the fabric has in response to heat and humidity changes. So those are some challenges, but it demonstrates this opportunity to bring together new types of engineering and things that we observe in biology to create new kinds of fabrics and new kinds of wearables that would be hard to do just using one of those systems. This is mushroom root, otherwise known as mycelium. It's this incredibly versatile material, and it's found all around us. It's fall now, it's almost October, and so all around us, every time a leaf falls and falls onto the ground, mushroom root decomposes it and forms it back into soil. So it's this very available process around us. And we know that we can shape the inputs that we put into the decomposition that happens with mushroom root and get very different kinds of outputs. Everything from something as light as foam to as durable and hard as concrete. It actually beats concrete in tests that compare the two, to something as pliable as leather. We can utilize mushroom roots and their decomposition process to create this by playing around with the inputs and things such as the nutrition and environment in which they're decomposing materials. And so, and what's really exciting about this too is it can offer this closed loop waste processing ecosystem where we can feed waste to the mushroom roots and they can break it down and produce these new kinds of materials we can use rather than having to produce them separately and create new types of waste as a byproduct. So I'll tell you a little bit about how people are using this. This is from a company called Ecovative. They're trying to reinvent plastics by using mushroom root. And their CEO talks a lot about how it's very easy today to get things shipped in 2 days or same day, and we have a lot more things being shipped to our homes and to our offices, et cetera. And you can return that easily, no problem. It's usually free. But the styrofoam that comes with your packaging takes 10,000 years to decompose. And so they're hoping that by using mushroom root and by putting it as kind of an intermediary in agricultural waste systems, they can create this sustainable type of plastic going forward. They also make bricks, and they collaborated with this architecture firm based in New York called The Living, which made this 40-foot structure made of 10,000 living bricks, and they were exploring ways that we can use mushrooms for building materials. They also tested the bricks against extreme wind, extreme heat, rain, et cetera, and they held up very well. And also, one of the interesting properties of mushroom root is it binds to other materials very easily, and you can also form it into whatever shape you want. And so this offers an interesting opportunity for construction going forward. And so here's another picture of the tower they built. Let's see. Here's another picture, and you can kind of see what the bricks look like. This is from a company called MycoWorks. They're exploring creating leather out of mushroom root and kind of changing the supply chain of leather to come from mushrooms. Last thing I wanted to talk about is this company called Mycotex. They're inspired by soft body systems, which grow in a modular way. And basically what they've been trying to do is create these modular parts of a textile by changing the inputs they give to the mushroom decomposition process. And so things like a collar or sleeves or ruffles or buttons are produced in different ways, and then they put that together and form an entire textile. So you can kind of see how they're growing some of those materials in the lab here. And so what's interesting here is just our ability to utilize this natural decomposition process to create new kinds of materials. So another group of researchers that's looking at how we can use naturally occurring processes in biological systems to create new types of structures is this research group led by someone named Neri Oxman at MIT Media Lab's Mediated Matter group. And so basically they were exploring ways that we can play around with what shapes the ways silkworms spin their cocoons and how we can learn from them and then utilize those as design elements that can create very different kinds of structures. So you can kind of see some silkworms spinning structure there, and then this is the full structure they ended up creating. And so basically they learned from the silkworms that things such as the light they were exposed to, silkworms have a guide thread, and so that things like the thickness of this thread or its orientation all shape the type of silk that's produced, and like the thinness and thickness and other qualities around it. And so they were able to take that and then use CAD and use different programs to design the structure they wanted to create. They created 26 polygonal panels with different guide threads, and they set 6,500 silkworms loose on it. And then over the course of a few weeks, they created this structure. And what's cool is actually the silkworms naturally deprecate by laying eggs and becoming moths and flying away. And so, and then the eggs they lay can either help reinforce the structure, or they can create up to, I think, 250 additional structures like this. And so yeah, they learned from the silkworms, and they took those inputs, they designed the structure they wanted to create, and then they put the silkworms back on it and let them create this structure. And you can see here another picture of how they're creating it. And so what's really interesting here is this is something that would actually be quite hard for a traditional 3D printer to do, to create something that's so multimodal and such different types of textures across an entire structure, and also even just to print something this large. But it's actually something that's quite easy for silkworms to do. And so, so they're exploring ways that we can expand our tool set that we use for architecture and design to include living systems. Besides that, they were explored ways that you can use silkworms and the type of output they're producing as a way to learn about the input, like to learn about the materials they're being guided by. And they're exploring uses of this in architecture, fashion, and also one that I'm really excited about, which is disaster recovery and relief efforts. Like, think about what if we could create these pseudo-structures and let silkworms create shelters for people. So there's a lot of interesting ways that they're exploring utilizing this for design and architecture, creating new materials of the future. Here's another picture. Another group that's looking at how we can look at the materials available to us to help us solve problems that we would otherwise have to create materials in a lab to do is this group led by a researcher named Andrew Pelling at the University of Ottawa. He has this interdisciplinary group of material scientists, stem cell biologists, physicists, software engineers, artists, and they all come together and try to think about what speculative augmented biology systems of the future might look like. And one of the areas they focus on is regenerative medicine. And so basically, regenerative medicine is this really exciting opportunity for us that as our bodies break down and our organs fail, we'll be able to grow organs in a lab that will either help repair our body, or we can replace organs in our body that are breaking down with these ones we're growing in a lab. And the ways that people are currently doing this are, one, either donated organs. I don't know how many of you are organ donors. I'm one. But people are taking those donated organs and either if that's the organ that someone needs, they're putting that in someone's body, or if it's not the organ that they need, they do something called decellularization, where they basically remove all of the living cells and this underlying polymer matrix that naturally occurs in the organ is present, and then they put stem cells in it, and they let the organ that they'd like to generate grow in that matrix, and that is successful. And then besides using donated organs to do that, people have also created this material in a lab, and so they grow organs on that. But both of those are kind of expensive approaches, and so Andrew Pelling and his team, they were wondering, like, nature provides us this almost endless variety of architectures and shapes and structures, and maybe we could find something that kind of mimics what we're producing in a lab that's already available to us in nature. And so they did, and they found apples. So Andrew Pelling's wife is actually a puppet maker, so she's very good at carving wood. And so basically what you see here is she's carved this ear shape in apples, and then they decellularized the apples, they put stem cells in it, and they were able to successfully penetrate the scaffolding and grow into organs that you could then integrate with living systems, including mice and humans. And they're very inspired by open source, and so they made sure when they were decellularizing the apples to only use available materials, easily available materials like dish soap. They also created this open source DIY incubator that is inspired by Andrew Pelling's dumpster diving, and so this is what you need to put the organs in to let them grow. And so, I wanted to mention that I have soybeans, mushroom roots, and apples as giveaways for after the talk, and I have instructions on how you can decellularize the apple and grow organs in it, as well as how you can ferment the soybeans, and how you can use mushroom root to create new structures as well. So I'll have that at the end. But so beyond this Apple project, Andrew Pelling and his lab are really interested in how we can look at the ways that cells adapt to environments that they didn't evolve to succeed in and are still able to succeed in those, and how we can push that to the limit and explore new kinds of biology and engineering with that. And how you can do genetic engineering without actually modifying DNA, but just by understanding how you can bring different biological systems together and help them integrate. So one of the next projects they're working on is asparagus. Asparagus looks kind of like what we're trying to do with regenerative medicine when creating spinal cords and also blood vessels. So they're exploring that as a new type of tool for regenerative medicine. Another set of companies that's exploring, and researchers who are exploring ways we can take this type of technology, such as decellularization, and specifically use our body to create new parts of our body outside of our body to put back in our body, or also to use our body to create new types of organs that we can then test out medicine and drugs that we'll put in our body, are this group of companies including Epibone, Tara Biosystems, Heprogen, and Emulate. So I'll talk about Epibone first. So they're exploring how you grow human organs outside the body. They started with bone because even though it has a complex shape, it's a single cell type, so it's kind of a good test case. And they were like, we can solve the shape issue through 3D printing, et cetera. And so what they do is when you have a bone defect, they take a scan of your defect, and then they take multipotent stem cells from your fat cells, which can grow into a lot of different types of organs, and then they create an incubator that allows the bone to grow exactly in the shape of the defect you have, and within about 6 weeks, bone tissue starts to form. And so you can see another picture of how they're doing that here, and beyond being a test case for exploring how we can grow organs outside the body using the body, it solves a specific problem. It's recovering when you have a bone defect and you're putting in new bone can be hard for a few reasons. First of all, your body might reject it. Second, it might not be exactly the shape that you need, and so fusing that together with your bone might be challenging. And then also you might introduce diseases or other things from a bone donor. So that's Epibone, another example of bone. This is a lung on a chip. This is made by a company named Emulate. So Emulate, along with Heprogen, which is trying to grow liver tissue outside the body, and Tara Biosystems, which is trying to grow mini hearts outside the body. What they're trying to do is help to push forward personalized medicine and drug development by mimicking the way your body is producing organs in development or in healing, and testing the way you'll respond to drugs by creating these isolated test environments. And so it's kind of this interesting concept around how we can bring together our understanding of how the body works with new types of technology and engineering to push personalization forward. Another company that's trying to grow mammal tissue in a lab is Modern Meadows. They're trying to be the first lab-grown leather company. So you can see some of their leather there. Initially, they were trying to grow cow cells in a lab, but this proved challenging for a few reasons. So they were trying to actually just grow cow skin instead of growing the entire cow just to produce leather. But this proved challenging because it required this nutrient-rich medium. They had to have fetal bovine serum to give it those nutrients, and because it was a nutrient-rich medium, their samples kept getting contaminated by things such as yeast. So then they were like, what if we can reframe the problem? And instead of trying to keep out the yeast from our samples can actually just be yeast. So they engineered yeast to produce collagen, which is the thing in skin that gives it its stretchiness and strength. And and then they were able to utilize a lot of the things that we have to produce yeast at industrial scale relatively cheaply through industries such as beer or even insulin production, which comes from yeast. So you can see an example of their samples here. And also by engineering leather in a lab, they were able to have a lot more control over the leather that's produced than they would have if it just came from a cow. They could play around with the thickness or thinness of the material, its stretchiness, et cetera, and kind of use those as design elements. And beyond that, they also weren't limited by things such as the size of the cow. They can produce these really large sheets of leather. So just another picture. This is their lab. Some equipment they use. This is what the leather looks like kind of in the middle of the process, and this is what it looks like when it's cured. So it basically looks like leather. And actually, starting tomorrow, they'll have their first sample on display, a shirt at the Museum of Modern Art in New York. So if you're in New York, tweet at me. I'll buy you coffee, and then we can go check out these shirts. Another material that we've known for a while that has these really powerful properties, but we haven't really been able to grow at scale and utilize those until recently is spider silk, which is actually just the silk found in a spider web. So I feel like when you describe spider silk, it kind of feels like you're describing Superman. It's stronger than steel, tougher than Kevlar, more flexible than nylon. It can expand up to 40% beyond its original length. And we've known about these things for a while, but we haven't really been able to produce it at a scale where it's been able to be impactful in industries such as medicine, architecture, or even creating more absorbent bumpers for cars, until recently. So people have tried to grow spider silk at scale in different ways. They tried to grow these industrial spider farms, but at some point they didn't really, like, the ecosystem didn't support itself, and basically spiders started to eat one another. Then people tried to engineer goats to produce milk that would create spider silk, and that sort of worked, but it ended up being kind of an expensive way to produce spider silk. So then they turned to yeast, different research groups, and so I'll talk about a few ways that people are utilizing yeast to produce spider silk. So this is from a company called Spyber, based in Japan. They engineer bacteria to produce fibroin. That's the protein that gives spider silk its properties. And so, and then they spin the output the bacteria generate into silk. And so you can kind of see what that looks like here. They're able to actually engineer a lot of kinds of silk that don't exist in nature because of their level of configurability around designing the bacteria that's producing the silk. So they're able to, like, optimize for strength or other qualities that they'd like to explore. And so they've been able to create 250 different types of silk, and they also are able to produce a new type of silk within 10 days. So that's pretty exciting. Here's a shirt they made, or dress, and then this is the moon parka they made in collaboration with North Face. This is from a company called Bolt Threads, based in San Francisco. They're also exploring growing spider silk in a lab. I think they ferment corn with genetically engineered yeast, and the output is this kind of spider silk material. And so they've actually created they've created, I think, over 2,000 types of new fabrics as a result of this process. Last example I wanted to talk about with spider silk is this project from students at the University of Pennsylvania. Spiderweb, beyond beyond holding prey, also filters out air pollutants. And so they created this living air pollutant that someone can wear, and a spider is weaving its web and protecting the wearer. Last thing I wanted to talk about related to this concept of scale is data storage and DNA. So we have a big data storage problem, and we're continually producing more data, and we know that DNA is actually the most efficient form of data storage that we've ever seen. It's able to store up to 215 petabytes of data in a single gram. And so researchers have been exploring ways that we can utilize DNA to store data. And obviously, it's going to be with us for a while, DNA, and it's quite versatile. And so in 2012, the first group of researchers were able to store data in DNA. It was a group of geneticists at Harvard, a guy named George Church and his colleague Sri Kasuri, and a research team around that. And they actually stored this book by the researcher George Church in DNA. But the method they used was pretty inefficient and didn't really utilize the full limit of what we're able to store in DNA. So more recently, researchers at Columbia University devised this algorithm called a DNA fountain, where they took a computer operating system, a computer virus, a film from the 1800s, and an information theory study by Claude Shannon, and they put it into one file, and they converted it to binary, and then broke it up into different binary strings, and randomly split those up into droplets and added these tags to help recombine it and then they sent it out to a company called Twist Biosystems, which I have a picture of here. And I think 2 weeks later, they got back this speck of DNA with their data. And the big advance there was that they didn't actually have any errors when they recombined the data. So that was a big step forward. Still, data storage in DNA is quite expensive. And people are hopeful that Microsoft, who's recently operationalized a goal of using data storage in DNA by 2020, somewhere in their organization will help push the cost down. Last interesting thing about that is we're exploring ways to lower the costs and increase the speed of the ways we read and write to DNA. And so we're trying to produce DNA in a lab using the same enzymes that we're producing it, using to produce it in our bodies. So all of these offer interesting ways of thinking about how we can expand programming today. Oh, hello. Okay. All of these offer new ways to think about how we can extend programming today to include different materials around us. And so when I was trying to understand, and the reason this is so important is because so many of the materials that people interact with are not the ones that we end up building for. So when I was trying to understand this concept around how we can extend the interfaces we build for, I got to this field of ubiquitous computing, which goes back 30 years ago. In 1991, Mark Weiser, who was then the CTO of Xerox PARC, published his vision for ubiquitous computing, which is this idea that computing shouldn't be something that takes us away from our environment, but it should become part of our environment. He wrote that the best technologies are those that disappear. They ambiently become part of our environment till we don't see them, rather than something that takes us away from the spaces that we're living in. And he also wrote that there's more information available at our fingertips during a walk in the woods than when we're using our computers, but one connects us to our environment and one takes us away from it. So when we think about biological systems and how we can utilize the different cycles and feedback loops and different attributes that occur in them to extend programming, I think that this can help orient us. So in sum, we can extend programming interfaces to include materials by bringing together biology and new types of engineering. And we can already develop for the broad range of interfaces that people interact with. And even the Internet of Things, which is trying to bring computing to materials around us, is still trying to put computers on things rather than reimagining things as computers. And so we can try to broaden our perspective of what a computer is and where we can do computing to incorporate a much wider range of materials. And so knowing what we know about what's possible, I want to know what experiences you want to design today with these materials. And I thought we could end with a moment of self-reflection. So think about the shirt and pants you're wearing or dress, the chair you're sitting on, the bag you're carrying, and just the materials you've interacted with today. What information do they have about you and your surroundings? How can we extend the experiences we design for to include those materials? So with that, would love to answer any questions or talk more about this topic. Like I said, I have some materials here that you can get started with programming. And if you go to this bit.ly link, you can find some resources for how to get started, as well as my presentation and speaker notes. Thank you.